Hello, I'd like to thank OIS for allowing us to present and to kind of reintroduce recombinant human lubricin to the community here. So lubricin is a protein originally discovered in synovial fluid and on the surface of articular cartilage, and was found to be the most lubricating component of synovial fluid. Since then, it's been shown that it's expressed at every interface in the human body, including the ocular surface, expressed by the corneal epithelial cells and the lacrimal gland. In addition to its lubricating functions, it's now been found that lubricin has quite a few biological mechanisms of actions. Particular to the ocular surface, it's a potent MMP9 inhibitor, as well as a TLR4 and NF-kappa B antagonist and inhibitor. So this is an image here of the corneal epithelial cells highly expressing lubricin. We also have discovered it's in the corneal endothelium, and importantly, it's downregulated in dry eye disease. You can see here on the images. In an in vitro model of an ocular surface on a chip, we found multiple biological signals, including reduction in TLR4 and NF-kappa B, and a broad variety of pro-inflammatory cytokines like IL-8, TNF-alpha, and IL-1 beta. But importantly, how does this translate to the clinic? Lubricin has been put into 3 clinical trials, early phase proof of concepts. This one here was a 40-subject phase 2 study comparing lubricin to the Vismed hyaluronic acid containing product. And it showed dramatic and statistically significant improvements against Vismed in multiple symptoms of dry eye and multiple signs as early as day 7. Here's just the Sandy frequency, but we also hit foreign body sensation, sticky feeling, blurred vision, photophobia, and the total symptom score. In addition to the symptoms, we saw multiple improvements in the signs of dry eye, including a greater than 50% reduction in overall corneal staining at 14 days, and a substantial improvement in tear film breakup time, as well as reduced erythema at both the eyelid and conjunctiva. For the first time here, we're presenting some data we did in a pilot study in post-refractive surgery dry eye, where we looked at patients 6 months post-LASIK or PRK who had persistent complaints of dryness on the ocular surface. And we saw here an evaluation of a low-dose lubricin compared against HA. We saw a very, again, profound response as early as 15 minutes, showing a greater than 30-point reduction in VAS, dryness, consistent across the course of treatment. And we also saw a dose response showing the 50 microgram per mil was superior to the 20, which was analogous to hyaluronic acid containing Vismed. We've also done quite a bit more research recently looking at the biological activity of lubricin. We did a study with Dr. Perez in his graft versus host disease model where we gave lubricin through an interperitoneal administration. So we're not looking at the lubricating function of lubricin, we're looking at its biological activity. And we saw a broad improvement in both the signs of ocular graft versus host disease as well as the systemic disorder, including signs like alopecia, body weight, and overall death. And that got us very excited about bringing lubricin back to the clinic for ocular graft versus host disease, which we'll talk about in a minute. We've also recently discovered that lubricin is highly expressed in the retina, which was news to us as we discovered it, and importantly is dramatically downregulated in patients with dry AMD. We ran a model of the sodium iodate AMD model, and we saw that, again, a systemic administration of lubricin, intravitreal injection of lubricin provided really substantial protection to retinal structure and function. Now, some of you may be aware that in 2017, we licensed the rights to lubricin to Novartis, and they took over and manufacturing and created a program they called ECF 843, which ran a very large-scale phase 2 study in 2002, which read out negative. Since we've gotten the program back, we've evaluated the molecule lubricin as they made it and the way we made it, and we found a number of large structural differences between the proteins and have published those results. Importantly, The size of their molecule was much smaller than our recombinant human lubricin. Their molecule was positively charged versus ours was negatively charged. And a broad way of looking at all the different activities saw substantial differences between the molecules. Since we got the program back in 2022, we've been focused on manufacturing a new batch that would be suitable for clinical trials, and I'm pleased to report that the batch has been released for clinical testing. The preclinical tox is clean, fill-finish campaign's complete, and we now have ethics approval to begin an ocular graft versus host disease study, which is set for first patient, first visit in December 2024. And so we're really excited to be back into the clinic and get this molecule hopefully back to patients and bring it forth. So thank you very much.